morning children. This is English video session for class 10 and the book is supplementary reader. The topic number 6, the making of a scientist. Richard E. Bright is now in his full focus towards the study of the butterflies, the differences between the monarch butterfly and the viceroy butterfly, how the viceroy butterfly copied the monarch butterfly so that they cannot be eaten by the birds, how they would develop. And what was the research which he did? He always felt that there is a great number of butterflies which are getting damaged during the by the beetles. But when he has put the beetles in the butterflies, nothing happened, and that became the greatest. Nothing happened becomes the greatest experiment, because the thought notion and the experiments proved that beetles are not a dangerous thing for the butterflies. Okay, now in this in his second year on high school, Richard E. Bright began to research that led to the discovery of an unknown insect hormone. Indirectly, it also led to his new theory on the life of cells. Okay? Now, the question he tried to answer was simple. What is the purpose of 12 tiny gold spots of the monarch pupa? So, this also he found out that there are tiny spots and the number is 12. Butterfly pupa is so small. In that, he has found that there are 12 dots of the pupa and everything are there only in the monarch. Okay, how did it happen? That is what he is finding out. Let's see. Yes, everyone assumed the spots were just ornamental, E. Bright said, but Urquhart did not believe it. Okay, so usually the designs which we find on the butterflies is more ornamental, decoration. But there must be something else to do this. So, to find the answer, E. Bright and another excellent science student first had to build a device that showed that the spots were producing a hormone necessary for the butterfly's full development. This project won E. Bright first prize in the county fair and entry into the International Science of Engineering fair. There he won the third place for zoology. He also got a chance to work during the summer at the entomology laboratory of the Walter Reed Army Institute of Research. As a high school junior, Richard Everett continued his advanced experiments on the monarch pupa. That year his project won first place at the International Science Fair and gave him another chance. Okay, that gave him another chance to work in the army laboratory. That gave him another chance to work in the army laboratory during the summer. So now he is going ahead in every spheres of work. He started experimenting. He found out that the 12 spots were producing hormone. That is development for the thing. It is nothing to do with ornamental. It is producing an hormone which is developed, which is helping them to the development of the butterflies full development के लिए वो help कर रही है. That is what he found out. Okay. So there are many. There he won third place for zoology. He also got a chance, and the entomology laboratory of Walter Reed Army Institute helped him. He was also given a second chance to work there. Now let's go. In his second year, he went to step further. He grew cells from a monarch's wing in a culture, and showed that the cells would divide and develop into normal butterfly wings scales only if they are fed the hormone from the gold spot. Remember, we already found that it is the hormone which is responsible for the full development of the monarch, this one. And who is doing this? Helping it for the monarch. It is done. We studied. Now what we are learning is what he found out. In the senior year, he went further. He grew cells from the monarch wings in a culture and showed that the cells, only if they were fed the hormone. Imagine you, the feeding from the hormone is not going, then the development stops. If they were fed for the hormone from the gold spot, okay, divide and develop into normal butterflies wing scales only if they were fed the hormone from the gold spot. So how does this cell develops? Again you read, in his second year he went a step further. He grew cells from a monarch's wing in a culture and showed that the cells would divide and develop into normal butterfly wings, okay, if they were fed the hormone from if they were fed the hormone from the gold spot. That project won first place for zoology at the international fair. He spent the summer afternoon graduating doing further work at the army laboratory and at a laboratory of the US Department of Agriculture. So what is he doing? He is growing cells from monarch's wing in a culture and showed that the cells would divide and develop into normal butterfly wings scales only if they are fed the hormone from the gold spots. Okay, yes. Now next, the following summer after his freshman year at Harvard University, Ebright went to the laboratory of Department of Agriculture and did more work on hormone from the gold spot. So this gold spot that is there in the monarchs really is a great puzzle for him and he goes on working on it. Using the laboratory's sophisticated instruments, he was able to identify the hormone's chemical structure. Imagine children, leave alone the hormone, the work which is doing on it. He also found the structure of that chemical, structure of that hormone. A year and a half later, during his junior years, Ebright got the idea for his new theory about cell life. He brought a new theory. It came while he was looking at the X-ray photographs of the chemical structure of the hormone. So there was something he found in the chemical structure of the hormone. When he saw those photos, Ebright didn't shout, Eureka, or even, I have got it. But he believed, along with his findings about insects and hormones, the photo gave him the answer to one of the biology's puzzles. What is that? One of the greatest puzzles. How the cells can read the blueprint of its DNA. And DNA is the substance of the nucleus in the cell that controls heredity. It determines the form and the function of the cell. Thus, the DNA is the blueprint of the cell. He brought new things which is already existing, but imagine. Eureka, he didn't do anything. He believed that along with his finding about insects, the photos gave him answer for one of the biology's puzzles. What is the puzzle? How the cell can read blueprint of its DNA. DNA is there, cell is there. How does the cell read the blueprint of the DNA? DNA is coming with the hereditary characters. But how come this will read that? Yes. Now, DNA is the substance in the nucleus of the cell. DNA, where is it found? Is it substance in the cell का nucleus में है. That is why it controls the hereditary. Okay, heredity. Again, let's go into the last most amazing discovery which he has done. Okay, Ebright didn't shout, but he found an answer for the what? Biology. Most puzzling thing of the biology. Okay. So, DNA is there, cell is there. We all know, we know cell is there, DNA is there. But how does this cell is reading the blueprint of DNA? It is only going with the thing of DNA. How is this going on? How do these two understand each other? Then they found out what is this DNA substance? DNA substance is in the nucleus, is a substance in the nucleus of the cell. So nucleus is the most important part in the cell which controls the hereditary. So where do you have cell? Cell ke andar you have the nucleus and this nucleus is controlled by the DNA and DNA is present there. Now understood children how things are getting read by each other. When it is inside, naturally it will read. Okay. It determines the form and the functions of the cell. That is how it determines the forms and functions of the cell. Thus the DNA is the blueprint of the life. That is why blueprint, it is showing the life. Understood children? Such a wonderful thing he has done. Yes, please read it again and again children. You all must learn this. Okay. Yes. Thank you children. Let's see in tomorrow's class.